So now we are back in the studio after listening to this very interesting talk by Dr. Robert Carter on 7 reasons to believe that Adam was real. So, Dr. Carter, are you with us?
I am. Can you hear me?
Oh, so thanks a lot for a very interesting talk, giving very convincing genetic evidence for a real Adam. So we have a couple of questions here, and the first one, you had these 7 reasons to believe that Adam was real, and I would say that reasons 2 and 3, they were based on the Y chromosome data, and it's very interesting. You mentioned that on one hand, the Y chromosomes of all men alive today are very similar. They only differ in, let's say, a few hundred letters, in spite of the fact that the Y chromosome is several million letters long, whereas the Y chromosomes of the chimpanzees are very different. So could you just expand on this a little bit more, in which sense the Y chromosome of the chimpanzee differs, for instance?
The human Y chromosome is notorious for having a lot of recombinations and duplications and inversions. There's if you just look from one man to the other, you see a lot of chromosomal differences. So therefore, the evolution has kind of expected the human and chimpanzee Y chromosomes also to be very different, at least after they started sequencing them. They said, oh, well, this thing changes really fast, therefore they're different. So it's kind of like a two-sided argument. They're not very surprised at the moment that we're very different, and I'm not very surprised at the moment that we're very different, because the Bible would indicate that we are. But that's the reason why there's so much differences between us in the evolutionary story. And yet, how do you tell? I mean, if you just line them up, they're incredibly different. So they could easily have been created different or not. Actually, I can't answer the question. Sorry.
Yeah, but I think it's very striking that human Y chromosomes are so similar, that they just differ in a few hundred letters, so to speak. So,
in fact, the Neanderthal Y chromosome has very recently been brought into the modern human fold. When they first got some pieces of the Neanderthal Y chromosome, it looked very different. But then they came out with some very high quality Neanderthal Y chromosomes, and they are human, clearly human, fully human, modern human. They fall beautifully within the realm of humanity. So Neanderthal, modern human, very similar Y chromosomes. Chimpanzees, radically different.
Yeah, that's so interesting. So for the second question, let's move on to your reasons number 4 and 5, and they are both based on non-sex chromosome and X chromosome DNA data. And if I understand it correctly, that your argument was that most of the genetic diversity we see among humans today for this kind of DNA was present already in Adam and Eve. So this is different from the Y chromosome DNA. And since only a small fraction of genetic differences among humans alive today are due to mutation, that is an argument that Adam and Eve lived recently. Is that a correct interpretation? That is, most of the genetic diversity we see today for non-sex chromosomes and X chromosome, that was put into Adam and Eve from the start?
There are about 10 million variants in the human genome that are found in our world populations. So maybe there's a section of chromosome number 1 and a person has an A or a T. Well, that A and T would be found, across the world. You, as an individual, probably carry about 3 million of those. If you take 4 or 5 people from any major world culture, you're pretty much going to get 95% or so of the world's genetic diversity. I mean, there's a lot of diversity within us, and it's spread around the world. Well, that could easily fit into Adam. But there's another set of variants, and those are the rare ones. Things that only appear in me or you or in a small people group or population. Those apparently are, because they're rare, it's highly likely that they're new, that they weren't at Babel, they weren't on the Ark, they weren't in Adam and Eve. They're new mutations, and most of those actually are a much higher proportion of those are disease-causing mutations. Now, the evolutionists would say, oh, well, natural selection hasn't had time to filter them out. And I would say, no, they weren't initially created. They've occurred more recently, and they're an evidence of a breaking system, not God-created diversity.
Yeah, that's so. It's so interesting. It's like we have a double argument: the low genetic diversity among Y chromosomes, in accordance with the biblical models, on one hand, and on the other hand, high diversity, which could be explained by created diversity for X chromosomes and non-sex chromosomes, apart from, as you said, the rare variants. So it's interesting that both of these aspects fit the biblical model. So then my third question is, well, an evolutionary biologist could say that, well, we have, like, if we have the evolutionary model where this is the human branch going backwards in time, and this is a branch going backwards in time from the chimpanzees, but if it would be the case that along the human lineage or branch there was a bottleneck from which only 2 people survived, that would be the correspondence of Adam and Eve, then we could expect the same kind of like Y chromosome and mitochondrial DNA data. But wouldn't that such a model in any case have a problem, because we would have more problems with inbreeding when the children of Adam and Eve marry and get children?
In the International Conference of Creationism paper that I talked about in my talk, where I showed the curves of diversity. The curves of diversity, and there's a lot of rare mutations and a few common mutations, and we use a chromosome 22 data to show that. And then we ran a bunch of computer models to show that a biblical model fits the data really well, an evolutionary model doesn't. That's one argument. The distribution of alleles fits the Bible beautifully. You can take 2 people out of that population, out of an evolutionary population, and kill off everyone else and have them restart humanity. We call that the evolutionary Adam and Eve. And you will get a very similar picture to the picture you get if you just have a biblical Adam and Eve. You can't hardly tell them apart, except what we didn't model was the effects of inbreeding and mutation. If you start with 2 God-created people that don't have any bad mutations, but have a lot of genetic diversity, there's no inbreeding effect. If you pull 2 random people out of an evolutionary population, they would have an excess baggage of deleterious mutations from their evolutionary history. If you have those 2 people restart the human race, you have a big problem. And the problem is that, what if one of those people was a carrier for hereditary blindness or sickle cell anemia or, any number of other genetic issues? You have a high likelihood of the population just crashing afterwards because of the mutation load.
Yeah, thanks. Well, that's such a strong argument that against such a evolutionary bottleneck of 2 persons because of this inbreeding effect, as you say. Because today, siblings are not allowed to marry and so on for the same reason. So, Göran, you had another question.
Yes, it's a question about mutation rates, because we calculations or measurements of mutation rates are used to confirm the age of mitochondrial Eve and Y chromosome Adam some 6,000/4,500 years ago. So, in your speech, you're arguing that there is a problem with mutation rates, that they may not be constant, and of course it must be so. But does those 2 views contradict each other, do you think? Can we still use the argument that Adam and Eve are so young, while still withholding that the rates may vary very much over time?
Yes and no. There's all sorts of ways where you can speed up mutation rates through radiation exposure, inbreeding, defective DNA repair enzymes, all sorts of ways to get mutation rates to go faster. It's really hard to slow mutation rates down because DNA is a fragile molecule. It's exposed to oxygen and water and cosmic rays. And so mutations will have a certain base rate. But also, you said initially that they used mutation rate to figure out the date for Adam and for Y chromosome Adam or mitochondrial Eve. That's not actually true. They do not use a modern mutation rate. They don't use a mutation rate that you can measure within families today. Instead, what they would do is they would estimate the genetic difference between humans and Y chromosomes in those areas, and then divide by the number of years we've been separated. So like 6.5 million years. That gives you a very slow mutation rate, but that's not actually something they measure in a laboratory. It's an extrapolation they calculate from the data. If you use the real world mutation rates, you will get a much more recent Y chromosome Adam and mitochondrial Eve. I don't think you can put a very accurate date on it because I don't think mutation rates are perfectly, I don't think mutation rates are constant over time necessarily, but you can't quadruple it. You can't multiply it by a factor of 10. You might be able to double it. You might be able to halve it or something like that. So within an order of magnitude, the current mutation rates fit a biblical picture. Is that exactly 4,500 BC? No, but that's not the nature of science. You're not going to get a number like that, but it's going to be within a range, and that range is a biblical range, and it's not an evolutionary range.
Yeah.
Did I say that okay?
But when I cited those 6,000, 4,500, then I was referring to the real measurements and not the hypothetical comparison between chimps and humans.
Okay.
So, but okay, that's I get the answer. Thank you.
Mm.
They also use archaeology to date things.
Yeah.
In Posnick's massive Y chromosome paper, which is pretty much a definition of the Y chromosome data, they're the people, the lab that submitted the Y chromosome data 1,000 Genomes project. They said that they used the peopling of the Americas as a sanity check. So the archaeological date for the peopling of the Americas is what they used for their calibration of their clock. Wow. That's not even genetic.
Yeah, I think this is so interesting, these laboratory recent estimates of which are more reliable of mutation rates. So I think we have time for a last question. I would just say that so much has happened within genetic research the last 10, 15 years. So do you think do you see any new particular trends that will further the creationist argument even more within genetics? Something that remains to be done, so to speak?
Well, the new trend right now seems to be ancient DNA, which should not even exist because DNA breaks down too quickly. Ancient DNA has allowed us to build really interesting patterns of migration and relatedness. Ancient DNA has brought Neanderthals into humanity, then Denisovans into humanity. I'm really expecting that's going to be the new trend as far as history goes. The thing is, I just read a paper this morning. I haven't fully read it, but I came across a paper this morning where they did this massive family tree, and they used ancient human DNA to root the branches in the tree, and they're still claiming that we all came out of Africa. So it's a new thing that needs to be analyzed. I mean, the older claims, right? You can't get that much diversity with Adam and Eve. We proved that wrong. The distribution of alleles is wrong if you start with Adam and Eve. Well, we proved that one wrong. And now the next one now is going to be, can you start from Babel and get the patterns we see in relatedness across the world? I don't know the answer to that yet because it's brand new and we have a lot of work to do. But I think ancient DNA is going to be the most interesting and most fun thing to talk about in the future.
That's so interesting. And welcome back, Robert, for the moderated discussion later on today. And thanks again for your talk.